Good afternoon, or more like good evening, everyone. Thanks for staying. And I'm Yue Qin. I'm currently a PhD student at Dr. Chadiker's lab at UC San Diego. And really happy today to meet all of you here. And hello to online folks. And we'll be sharing you like our latest work. Please check out our paper for more details if you're interested in the talk. So let's get started. I'm sure all of you know the exciting news that we finally have a complete genome mapped and assembled. But with that, it's like we know like what roughly are the proteins in our cells. However, a big missing piece, I say like we're just scratching the surface in knowing this. It's like how these proteins, how do they come together to eventually form the complicated structure of a cell? Especially if you consider the cell structure, it spans multiple scales and have different organization that naturally arise at different scales, like protein complexes, pathways, organelles, and not mention the cell. So how do we study this, like map this complete structure? And in the past, like biologists, scientists have been developing very exciting technologies, namely here like two popular ones are protein immunofluorescence imaging and protein-protein interaction. So for protein-protein interaction network, you often see like it as a massive hairball view here at the lower left corner. And usually this protein-protein interaction, it tells you a very local information of the protein. If you can see, like it usually informs you about the protein physical associations or protein complexes that arise at a few nanometers to tens of nanometers. On the other hand, imaging is a very popular method to assay the protein localization at a much broader scale. It gives you a global distribution pattern of this protein inside the cell. However, despite their distinct format and quality and resolution, they actually have one information that is shared among them. That is that they both give you a kind of coordinate of this protein inside the cell. So it might be more obvious for imaging data that you just like kind of assign a coordinate to the protein based on its relative localization to cellular landmarks. For example, the blue of nucleus and the red staining cytoskeleton, as I'm showing here on the lower right, which is a typical immunofluorescence imaging that you see in most studies, and it's the kind of it's the exact data that we're using in our study. But like if you think about protein-protein interaction, it gives you actually the coordinate of the protein, a more abstract, a local coordinate is that it puts each protein inside the cell based on who their nearby interactors are. And so then like once we have a coordinate of a protein, can we then start to measure the pair distance between them? And once we have distance, can we then like do pan resolution community clustering, like clustering of the protein-protein distance at different scales and different resolutions to find the protein communities that naturally arise at different at this multi-scale structure. So to pilot our method and to study it, we collaborated with the Bioplex team and the Human Protein Atlas group, as they each is the largest database within each data modality respectively, namely affinity purification mass spectrometry and protein immunofluorescence imaging. And one nice thing about these two large-scale efforts is that despite them like studying different cellular contexts and different cellular backgrounds, there is one shared cellular context between them, which is HEC293, a human embryonic kidney cell, and it's exactly the cell type that we developed our approach in. So to start with, our approach consists of 3 steps. First step, can we give a coordinate to the protein? And the way we do that is we embed each protein from each data modality separately, meaning that after this, each protein has one image embedding, capturing its global location pattern inside the cell in reference to the cellular landmarks, and another protein-protein interaction embedding that captures its local neighborhood information inside the cell. So with that, we then like train a random forest regressor to let it to learn the general measure of protein-protein distance from the embedding of a pair of proteins, and from which, by applying high-resolution community detection, this is how we assembled our multi-scale subcellular hierarchy. And just a bit more on this pan-resolution community detection. So imagine you have a network with edges denoting the protein-protein distance, and I'll say, okay, only show me the edges that represents the strongest, the closest distance or the strongest association. That is why you see at the bottom here, edges that connect small protein complexes is representing strong association between the proteins, and these are usually like representing small protein complexes. But as you lower the stringency, you're gradually bringing edges that represent longer distances, as you see show here, like seeing in the middle, the red edges, and even lower the stringency, you will start to see like those purple edges on the top. And through this way, you gradually bring in more edges and find larger and larger protein communities that contain the previously identified smaller ones. So by applying this strategy on all pairwise protein distances, this is how we derive the version 1.0 of our multi-scale integrated cell music. So just a little walk through this, like music here is also represented as a network, however, very different from the protein-protein interaction network that you have been seeing likely most often and what I've been talking about. So here, each node in the music map represents a protein community instead of a single protein. And the node size denotes the number of proteins annotated to this community, and also the color of the node is showing whether this community is previously known or not. With gold showing that it's already being annotated, or with purple showing that it's putatively novel, and we should dive into it to see if we can find cool novel biological insights. And so the edges between the nodes in this case represents a protein containment relationship. So meaning that if you look top here, very simple example, cytoplasm and nucleus, they're contained by the cell. And also if you notice, I'm not sure if the figure is clear enough to show, some communities actually have multiple parents. So this is thanks to our special clustering method, allowing us to capture, to assign like communities to have overlapping members, thus allowing them to not only overlap proteins, but have multiple parents, the better capturing the biological dynamics such as pleiotropy or multi-localization of the proteins. And also, just let me further remind you that if you kind of imagine, zoom into each node, you will see a protein-protein distance network behind each node, as I've been previously showing you, and as I'm putting here on the left here. So, like with these protein-protein distances among its members, this is how it enabled us to estimate its physical size for each of these cellular communities. And this physical size is determining its like hierarchical layout on our music map, and is also annotated by the size letter here on the rightmost of the slide. So how well do we do, as I mentioned in slides? So here we compare music prediction to what's actually being annotated experimentally in the literature. So on this scatter plot, each dot represents a subcellular component, and the x-axis shows the music predicted physical size for that component, while the y-axis shows what we actually collected from literature, demonstrating that we indeed accurately sizes and captures biological systems across a wide range of scale. And also just to demonstrate what kind of biological insight you're able to derive from such a map. Here is one of a few examples that we've explored in our study, which we found like RPS3A, by name you could probably tell it's an ribosomal subunit protein. It's actually strongly associated with 3 other known splicing regulators. So this makes us wonder, like, why a ribosomal subunit protein is there hanging out with 3 splicing regulators? Does it have some unknown splicing functions? And this inspired us to collaborate with our colleague, Dr. Jin You's group here at UC San Diego, to together carry out an enhanced UV cross-linking precipitation assay, ECLIP. So just briefly on how ECLIP works. So in the cell, you have RNA binding protein denoted by RBP here that binds to different RNAs, and you UV cross-link their interaction, and then by pulling down the RNA binding protein using specific antibody, you're able to identify the RNA species that are bound by that protein as well as their binding sites. So with this binding sites, this enabled us to generate such an ECLIP profile, which characterizes, like, for each RNA binding protein, what is the profile of its like binding spectrum inside the cell. And as you can already see, RPS3A has a very similar ECLIP profile to that of canonical splicing regulators, but very distinct from canonical ribosomal proteins. That may be just anecdote. Let's do it globally. So we clustered RPS3A's ECLIP profile to all the publicly available ECLIP profile available online. And so we found that indeed RPS3A robustly clusters with RNA binding proteins that are specifically enriched for splicing regulation. And with that, I would like to conclude and thank my advisor, Trey, and also Emma, who co-supervised this work, and also like all my wonderful collaborators. And also just on a personal note that I'll be starting as a fellow at a new machine learning center at Broad Institute, Eric Wendy Schmidt Center. I'm very excited about this transition, and stop and say hi if you're in the area. With that, thank all of you for your attention.